What if I told you that a multi-million-dollar, ultra-high-resolution genetic map of a cancer tumor could be, like, perfectly replicated for free?
Right, using literally nothing but a black and white sketch from the 19th century.
Exactly. It sounds like science fiction, but today we are doing a deep dive into a groundbreaking AI model called Path2Space.
Yeah, this was just published in the journal Cell in July 2026, and it is completely upending how we understand and treat breast cancer.
It really is. So to really grasp why this is so revolutionary, I want you to imagine you're trying to understand a massive, complex city. You want to know its economy, its vibe, how it functions.
Right.
Standard genetic sequencing, what biologists call bulk RNA sequencing, is a bit like taking all the buildings, the coffee shops, the banks, the apartments, and tossing them into a giant blender.
A very expensive blender, but yeah.
Right. And then testing the resulting smoothie, you analyze it and say, okay, 10% of this city is made of coffee shops.
Which, I mean, that is useful information.
It is. But it tells you absolutely nothing about where those coffee shops are. Are they clustered downtown? Are they next to schools?
And that's a huge problem because in biology, just like in real estate, location is everything. You lose all the geography and the context with that blender approach.
Exactly. So to get the actual map, science developed something called Spatial Transcriptomics, or ST.
Yeah. If the blender is the smoothie, ST is your multi-million-dollar satellite map.
Right. It shows you exactly where every coffee shop is relative to every office building. But here's the catch. That satellite map is incredibly expensive. We are talking prohibitively expensive for routine use in large clinical trials.
Let alone everyday hospital care. It creates a massive bottleneck.
Right.
We have the technology to see the map, but we just can't afford to run it for the thousands of patients who actually need it.
And that is exactly the problem Path2Space solves. So we're going to unpack exactly how this AI works.
Yeah, it's not magic, though it really sounds like it. It's an incredibly meticulous feat of engineering.
So let's start with the blueprint, right? How does this AI actually read the unseen? Let's establish our baseline here. What is the cheap sketch the AI is using as its starting point?
Well, the sketch is a standard H&E slide.
H&E?
Yeah, H&E stands for hematoxylin and eosin. If you've ever seen like a picture of tissue under a microscope in a biology class or a hospital, those classic pink and purple cells.
Oh, right. Yeah, everyone's seen those.
That's an H&E stain. Hematoxylin is a dye that stains the cell nuclei, the control centers, a dark purple, and eosin stains the cytoplasm and the surrounding matrix, a bright pink.
Okay, so it's a very basic technique.
Super basic. It was developed about 150 years ago. It's ubiquitous, and it costs almost nothing to produce.
Okay, so we have a cheap pink and purple image.
Yeah.
And what is the expensive satellite map it's trying to replicate?
That's the Visium Spatial Transcriptomics technology. Visium takes a highly delicate slice of tissue and measures the gene expression, meaning it counts exactly which genes are turned on or off in these tiny circular spots.
How tiny are we talking?
Each spot is about 55 micrometers wide.
Wow.
Yeah, to picture that, it's roughly the width of a human hair. But as we mentioned, doing that across thousands of patients is just way too costly.
All right, so how do we get from a 150-year-old pink and purple dye job to a high-res map of invisible genes?
It happens in 4 highly specific steps.
Okay, put them on me.
Step 1 is tiling. The AI takes the high-resolution digital image of the H&E slide and digitally chops it up into a grid of tiny 80-micron square tiles.
So it's breaking the big picture into puzzle pieces.
Exactly. And it intentionally centers these square tiles exactly where those expensive Visium circular spots would normally be placed.
Okay, so now we have a digital pile of thousands of tiny pink and purple squares. What's next?
Step 2 is normalization. Think about how many different hospitals exist, right? They all use slightly different dye manufacturers, different slide scanners, different lighting.
Oh, sure. So the colors wouldn't match perfectly.
Right. If you don't fix that, the AI might think a darker purple means a different type of cancer, when in reality, the lab technician just left the dye on for an extra 10 seconds.
That makes total sense.
So to fix this, they use a mathematical algorithm called Marchenko's color normalization.
Marchenko's color normalization. It's like applying a standard color correction filter to an entire batch of photos on your phone.
Right.
Right? So they all share the exact same lighting and color palette. You're removing the human error.
That's a perfect way to put it. It completely decouples the intensity of the stain from the actual physical structure of the tissue.
Got it.
Once the images are normalized, we hit step 3, which is feature extraction. This is where the heavy mathematical lifting happens.
Brace me.
The researchers employ a foundation AI model called C-Transpath. This model has already been trained on millions of pathology images. It looks at our 80-micron tile and extracts its visual essence, basically squishing that complex image down into a 768-dimensional mathematical vector.
Okay, hold on. I need to pause you there. 768 dimensions.
Yes.
My brain literally stops conceptualizing at 3D. What does a 768-dimensional mathematical vector actually mean in this context?
It's a great question. Think of it like describing a person's face to a sketch artist. You don't just say, they have a face.
Right.
You break it down into features. The distance between the eyes, the sharpness of the jawline, the exact angle of the nose. CTransPath does that for cancer cells, but it uses 768 distinct visual parameters.
So it's incredibly detailed.
Insanely detailed. It measures the texture of the pink matrix, the density of the purple nuclei, the jaggedness of the cell borders, the white space between them. It turns the entire visual architecture of that tiny square into a list of 768 numbers.
Okay, so it turns an image into a highly specific numerical barcode of shapes and textures.
Yes. And that brings us to the final hurdle, step 4, the MLP, or multi-layer perceptron.
Which is a type of neural network, right?
Exactly, a classic artificial neural network. The MLP takes that barcode, those 768 numbers, as its input. It passes them through a hidden layer of mathematical weights and balances, and it outputs predictions for the exact expression levels of 14,068 individual genes for that specific tiny tile.
See, I have to push back here because this still sounds like a magic trick.
I know, it does.
If the AI is literally just looking at a barcode of shapes, how densely packed the cells are, how jagged the borders are, how can it possibly know which of 14,000 invisible genes are turned on? There's no physical DNA or RNA in a photograph.
It's a brilliant deduction based on a core biological rule. Form follows function, and function is driven by genes.
Okay, give me a concrete example. How does shape equal gene expression?
Let's look at cell density. If the AI sees a tile where the purple nuclei are massively crowded together, practically climbing over each other, it knows those cells are multiplying recklessly.
Right, because that's what cancer does.
Right. And what causes reckless multiplication? Specific proliferation genes like Ki67. So the AI learns that the visual pattern of extreme crowding strongly correlates with the Ki67 gene being turned on. It learns the hidden visual fingerprints of the genes.
Ah, I see. So it's not looking for the gene itself. It's looking for the architectural consequence of that gene being active.
Exactly. And the researchers actually made a fascinating discovery about how to teach the AI those consequences.
Oh.
Yeah, when training the model, they had to feed it the true gene expressions from the expensive Visium maps so it could learn. But the format of that data mattered immensely. They found that training the AI on the log-transformed raw counts of the genes worked vastly better than using a widely accepted normalized ratio called CPM, or counts per million.
Wait, why is that a big deal? Isn't CPM the standard way to clean up data in biology?
It is. CPM is standard practice to smooth out technical variations, like how deep a sequencer looks into a cell. But the AI completely rejected it. When they fed the AI the smoothed over CPM ratios, its performance tanked.
Wow. I love this detail because it reminds me of a detective investigating a complex crime scene.
How so?
Well, if you're the detective, you don't want the neatly typed, heavily edited, summarized police report. You want the raw, unedited, gritty security footage. The AI needed the messy, raw count data to properly learn the complex, subtle signals of the cells.
That is a perfect analogy. It proves that sometimes human attempts to neatly clean data before feeding it to an AI actually strip away the vital, subtle biological signals that the neural network desperately needs to make its connections.
So before we move on, let's just do a quick recap.
Sure.
We take a cheap H&E slide, we cut it into 80-micron tiles, color correct them, compress their visual architecture into a 768 number barcode, and a neural network decodes that barcode into 14,000 predicted genes.
That's the core of Path to Space.
It's incredible. And for you listening, it brings up an interesting question. When you look at a complex problem in your own life or work, are you looking at the blended smoothie, or are you looking for the spatial map? But even with the raw data, spatial transcriptomics is notoriously messy. How do they handle the inherent chaos of biological data?
That leads us to the secret sauce, really. An engineering trick that pushed Path to Space ahead of the pack called spatial smoothing.
Spatial smoothing, how does that work?
Well, once the AI predicts the gene expression for a specific spot, it doesn't just lock that prediction in. It looks at the immediate neighbors of that spot specifically, all the spots within a 200 micrometer radius.
So how many neighbors is that usually?
On average, that encompasses about 8 surrounding neighbors. The model then averages its predicted gene expression for the central spot with those 8 neighbors.
Oh, I see. So it checks its work against the local environment.
Exactly.
It's like cleaning up a grainy photograph. If you have a picture of a clear blue sky and one single pixel in the middle is pure, bright white, but the 8 pixels surrounding it are dark blue, that white pixel is almost certainly just static or camera noise. By averaging them together, you smooth out the technical glitch and reveal the true blue sky.
Precisely. In spatial transcriptomics, you often get something called dropout, where a gene is actually there, but the sequencer randomly fails to detect it. The spatial smoothing acts like noise-canceling headphones for this genetic dropout.
It dampens the random technical errors.
Right, and it amplifies the true biological signal.
And the results were pretty staggering.
Right, they really were. By applying this spatial smoothing, the number of highly predictable genes shot up from 1,376 to 6,629.
That's a massive jump.
It's huge. Because of this, Path2Space significantly outperformed 21 other state-of-the-art AI models that tried to do the same thing, like EGNV2 and Bleep.
But the part of the paper that genuinely blew my mind wasn't just beating other models. It was what this AI could do with historical data. The archival breakthrough. Can you explain the FF versus FFPE dilemma?
This is arguably the most impactful part of the study for everyday medicine. The AI was trained entirely on fresh frozen tissue, or FF.
Okay.
That's tissue that is immediately snap frozen with liquid nitrogen seconds after a biopsy. It preserves the RNA beautifully, but it's expensive and logistically very difficult for hospitals to do routinely.
Right.
But when the researchers went to validate their AI on external data, they fed it FFPE tissue, and it worked just as perfectly.
Let's define FFPE for the listener.
FFPE stands for formalin-fixed paraffin-embedded. This is the standard, everyday chemical preservation process used in nearly every hospital in the world.
Where they put it in wax, right?
Yeah, the tissue is soaked in formaldehyde and embedded in a hard block of wax.
And why is the AI working on these wax blocks such a massive breakthrough?
Because extracting high-quality RNA from decades-old wax blocks to run an expensive Visium spatial map is nearly impossible. The formaldehyde chemicals severely degrade the genetic material over time.
Oh, I see.
But there are vast historical archives of patient data sitting in hospital basements globally, locked in these wax blocks. Millions of documented cancer cases where we know exactly how the patient responded to drugs over 5, 10, 20 years.
Wait, I see where you're going with this. The AI doesn't need the degraded RNA.
Nope.
It only needs the image of the physical structure, the shapes, and the pink and purple architecture which the wax preserves perfectly for decades.
Exactly. Because Path2Space works on standard FFPE images, scientists can now unlock spatial data from millions of historical cases. It is literally time travel for cancer research.
Wow.
We can retroactively generate multi-million-dollar molecular maps for patients who were treated 20 years ago and correlate those maps with their long-term survival data.
So just to recap this section really quickly, averaging neighboring spots, spatial smoothing, acts as a noise-canceling headphone for genetic data, right?
Yep.
And this allows the AI to unlock decades-old wax-preserved tissues. Which makes me wonder, how often do you apply a smoothing filter to your own information intake, checking a single noisy data point against the neighbors around it to find the truth?
That's a great way to think about it.
So that is awe-inspiring. We now have this incredibly powerful tool that can perfectly map a tumor for free. The next logical question is, what does that map actually reveal about the cancer's behavior? When they unleash this AI on a massive data set, what did they find?
Well, they found that tumors have highly distinct neighborhoods, and those neighborhoods dictate whether a patient lives or dies. The researchers took data from 976 breast cancer patients from the Cancer Genome Atlas database.
Okay, the TCGA database.
Right. They broke those 976 slides down to 6.7 million tiny tissue pseudospots.
6.7 million spots. How does a human brain, or even an AI, make sense of that astronomical amount of geographical data?
They used an algorithm called Spatial, which stands for Spatial Graph Convolutional Network.
Spatial.
Yeah. It sounds complex, but the concept is intuitive. Imagine a social network where the algorithm looks at who you are, but also who your friends are to categorize you.
Right, like Facebook suggesting groups.
Exactly. Spatial does that for cells. It looks at a spot's predicted gene expression and its physical proximity to other spots and groups them into coherent geographic zones.
So it's finding the biological equivalent of the financial district, the residential suburbs, and the industrial zones within the tumor.
Precisely. Across all 976 patients, SPAD-GCN identified 11 distinct types of these zones, which they called spatial clusters. And based on the specific ratios of these 11 clusters, they found that all breast cancers fundamentally fall into 3 distinct macro categories.
And they call these SpatioTypes.
Yes, SpatioTypes.
I love a good mnemonic, so let's use PII to remember these 3 breast cancer SpatioTypes: proliferation, immune modulated, and immune inactive.
Good idea.
Let's break down the P first. What is the proliferation-enriched neighborhood like?
The proliferation-enriched spatiotype is dominated by clusters 9 and 11. Biologically, proliferation means rapid, unchecked cell division.
So it's growing fast.
Very fast. The map shows dense areas of cancer cells aggressively multiplying. Clinically, this spatiotype is associated with higher disease stages and more advanced tumors.
Okay, then we have the first I, immune modulated. From what I understand, if you have to have a tumor, this is the neighborhood architecture you want.
Relatively speaking, yes. The immune-modulated spatiotype features high levels of tumor-infiltrating lymphocytes, or TILs.
TILs.
Right. In practical terms, this means your immune system's army has successfully breached the tumor's borders and is actively inside fighting it. The map is heavily populated by cluster 5, and patients with this spatiotype robustly predict better disease-free survival.
Which brings us to the final I, the immune inactive spatiotype. This is the worst prognosis. What does this map look like?
It is a highly aggressive, hostile environment. The immune inactive spatiotype is dominated by clusters 2 and 4. In this architecture, there is almost zero immune presence.
Oh, wow.
Yeah, the cancer cells are incredibly dense, and there are massive spikes in oncogenes, the specific genes that drive cancer growth.
It's like the tumor has built a geographical fortress that the immune system's army simply cannot penetrate.
That is a perfect way to visualize it, and the clinical impact is severe. The researchers found that having this specific immune inactive spatiotype essentially doubles the hazard ratio for a bad outcome.
Yeah, let's make sure we understand that. When you say it doubles the hazard ratio, do you mean it's twice as deadly regardless of other factors?
Yes. Even after adjusting for the patient's age and the stage of the tumor, meaning even if the tumor is caught early and is relatively small, if the geography of the cells is immune inactive, the risk of a poor outcome doubles.
That's terrifying, but also incredibly useful to know.
It completely reframes how we view a biopsy. We aren't just looking to see if cancer cells are present anymore. We are evaluating the structural barriers and the socioeconomic status of the tumor's neighborhoods.
Let's do another quick recap. Using the AI, researchers discovered all breast cancers fall into 3 spatiotypes: PII, proliferation-enriched, immune-modulated, and immune-inactive.
Right.
If your immune system is a localized army, the immune-inactive tumor has built a fortress the army can't penetrate. What hidden fortresses might exist in the data you analyze every day?
That's the real question.
And understanding the structure of those fortresses is exactly how we figure out how to breach them. Which leads us to the most high-stakes part of this research, using this map to predict the future of treatment.
This is where Path to Space moves from an observational tool to a life-saving clinical application.
So let's set up a real-world clinical scenario. A patient comes into the hospital, and they are diagnosed with HER2-positive breast cancer.
Okay.
The standard medical protocol is to give them a highly targeted chemotherapy drug called trastuzumab.
Right, which is also known commercially as Herceptin.
Exactly. But we know it doesn't work for everyone. Usually doctors just look at that bulk blender smoothie sample to see if the overall level of the HER2 protein is high.
But as we've established, the smoothie misses the map. Just because there's a lot of HER2 in the blender doesn't tell the doctor how it is arranged in the tissue. So the researchers invented a brand new metric specifically to analyze the map. They called it SPAND.
SPAND. What does that stand for?
It stands for Spatial Neighborhood Diverseness.
Spatial Neighborhood Diverseness.
Yeah.
How does the SPAND metric actually calculate that diverseness?
It relies on a mathematical concept called Moran's eye.
Moran's eye.
Yeah, Moran's eye is a measure of special autocorrelation. Simply put, it calculates clumpiness. SPAND looks at a tiny 100 by 100 micrometer neighborhood in the tumor and measures how mixed up or heterogeneous it is.
Okay, so if I have a tumor where all the HER2 positive cells are clumped tightly together on the left and all the HER2 negative cells are clumped on the right, what is the score?
That would be a highly segregated neighborhood, which results in a low span score.
Got it.
But if you have a checkerboard situation, a chaotic mix of HR2 high cells sitting right next to HR2 low cells, that is a highly heterogeneous neighborhood, resulting in a high span score.
Now, this creates a massive logical paradox for me, and I really need you to explain this.
Okay, sure.
Trastuzumab is a drug literally designed to target and attack HR2 cells. Logically, wouldn't you want a tumor to be 100% perfectly solid with HR2 cells so the drug has a target on every single cell?
Yeah.
Why does a high span score, meaning a heavily mixed checkerboard of high and low HR2 cells, actually predict a better response to the drug?
It is a brilliant biological paradox, and it all comes down to the precise mechanism of how trastuzumab works.
Okay.
Trastuzumab doesn't just poison the cancer cell directly. It is an antibody. It binds to the HR2 receptor on the cancer cell, and the tail of the antibody sticks out.
Like a flag.
Exactly like a flag. This flags the cancer cell for the immune system, triggering something called FC receptor-mediated immune cytotoxicity.
Okay, let's unpack that. FC receptor-mediated immune cytotoxicity.
Think of the trastuzumab drug as painting a laser target on the HER2 high cells. Natural killer immune cells patrol the area, see that flag, and release chemical grenades, things like perforin and granzymes, to destroy the flagged cell.
Okay, so the immune system's artillery blows up the high HR2 cell. But what about the low HR2 cell sitting right next to it?
This is the key. Chemical grenades don't have perfectly clean borders. When the immune cell detonates its payload to kill the high HR2 cell, the blast radius takes out the cells immediately next to it, regardless of whether they have the HR2 flag or not.
It is biological collateral damage.
Exactly. In biology, this is known as the bystander effect.
Wow.
Now, apply this back to our span score. If the tumor was perfectly segregated, a low span set with all the high HR2 cells on one side, the immune system would wipe out that half.
But it would completely ignore the low HR2 side.
Right, leaving half the tumor alive to grow back and kill the patient.
But if the tumor has a high span score, if it is highly mixed like a checkerboard, the immune system targets the high cells, and the resulting crossfire takes out the entire neighborhood. The cancer's own chaotic mixing becomes its fatal vulnerability.
Precisely. And the clinical data backs this up phenomenally well. Using just the cheap H&E slide, Path2Space calculated the span metric and predicted pathological complete response, meaning the tumor was entirely eradicated by the drug with up to 83 to 90% accuracy across multiple independent clinical trials like TransNeo and Impress.
That is astounding.
Yeah.
The AI is looking at a virtually free slide, analyzing the spatial mixing, predicting the immune crossfire, and doing it with greater accuracy than $1 million bulk RNA sequencing lab.
Yeah, and they also found that cluster 4 from our SpatioTypes was the strongest predictor of a poor response to these treatments, giving doctors a massive red flag before they even start therapy.
So quick final recap. SPAN measures spatial mixing. High HE2 mixing allows the immune system to create a bystander effect, killing untargeted cancer cells. In biology, heterogeneity turned out to be a vulnerability for the cancer. In your own teams or projects, how does the precise spatial arrangement of different elements change the outcome?
It is a profound paradigm shift. We are moving from asking, what is this tumor made of, to asking, how is this tumor organized, and how can we use that organization against it?
It really is. We've covered an incredible amount of ground today. We started with an inexpensive 150-year-old technique of staining tissue pink and purple.
And by applying foundation models, multilayer perceptrons, spatial smoothing, and topological math like Moran's eye,
Path2Space extracted invisible genetic blueprints. We discovered the PII SpatioTypes of breast cancer, and we proved that the precise microgeography of cells can predict the bystander effect of life-saving drugs. The blender gives you the ingredients, but the map gives you the meaning.
If I can leave you with a final thought to mull over, Path2Space definitively proves that the deepest biological secrets aren't just hidden in what things are made of, but in how they are arranged.
That is very true.
It forces us to question our own perception. If a simple neural network can look at the architecture of a few stained cells and deduce 14,000 invisible genetic variables, what other invisible truths are hidden in the plain sight of our everyday physical world, just waiting for the right lens to decode them?
Take a look around your environment today. What invisible networks are shaping the reality you see?